Hi, everyone, and welcome back to part 4 of this lecture about the interpretation of estimates of heritability from SNPs. So in this part, I will talk about a famous problem that has been termed as the missing heritability problem. And I will also talk about biases in heritability estimation and how we could solution those biases. So starting with the missing heritability problem, so this is has become a very, well, it was a very famous problem about a decade ago. And that problem came from contrasting how much trait variance or risk was explained by variants that are detected in Genome-Wide Association Studies. So a handful of variants which were detected at that time, and how much I said a decade ago is actually much further than that. But how much those variants were explaining the trait as opposed to how much heritability was expected from family studies or pedigree studies that are looking at the resemblance between close relatives. And there was a big difference between those numbers. For example, for height, the handful variants detected at the beginning of 2010 or the end of 2010, where it was about 5% variance, height variance explained, whereas the heritability was expected to be 80%. And so that big difference worried, well, to some extent, the community in sense of asking what exactly are we missing, and that was termed as the missing heritability problem. So in order to understand that disconnect we may have between heritability estimate obtained from pedigree analysis or those obtained with a handful of SNPs, we have to recognize that relatedness varies across the genome. If you calculate the GRM from one part of the genome, and you compare it with the GRM calculated with another part of the genome, which is not overlapping, although they will be correlated to some extent, and in particular if the sample that you're using contains close relatives, but in general, those GRMs will be largely uncorrelated. And so that has an important implication, because it says that if the SNPs that we use to estimate heritability do not reflect well enough the degree of genetic resemblance at the causal variants, so the variants which are causing the traits that we analyze, then we should only expect to have a partial estimation of heritability, and which can be seen as a bias. And so this is what is expressed here in this inequality, which essentially shows H-square GWAS is how much variance is explained, the proportion of the variance is explained by SNPs detected in the GWAS. The SNP heritability here is an estimate that we can have irrespective of the degree of significance of those variants in the GWAS. If we just take as much SNP as we can, how much those SNPs explain, if we put them into a GRM, we get that estimate here, and that's H-square is the total heritability, which we could have essentially estimated. Well, either from pedigree studies or either if we had relatedness measured directly at the causal variants. And so that's kind of the core of the issue. So where we are at the moment, I'm just going to also introduce some funny exotic terms which have been populating the literature, the GWAS literature, but starting with the missing heritability, which I've already defined. So it's essentially the difference between that quantity here, the total heritability, and the heritability explained, or the proportion of variance explained by SNPs detected by GWASs. So we can see that as GWAS sample size increases and we detect more, that this difference in SNP missing heritability is shrinking. There is another quantity that people have been terming as hidden or hiding heritability, which is essentially the difference between H-square SNPs and H-square GWAS. And so what we know based on a number of publications, in particular seminal work from Jian Yang in 2010, is to show that as we increase sample size, if we set constant the set of SNPs over which we are testing associations with our phenotype, as we increase sample size, we expect that the still or the hidden heritability to go to 0. And there is another part that is called the still missing heritability. And essentially that still missing heritability is acknowledging the fact that, well, with SNPs or with imputation or with the technology that we have, we may not be able to assay properly all possible causal variation for the trait that we're analyzing. And therefore, there will be a gap between H-square SNPs as based on an arbitrary set of SNPs. No, we can take it to be as large as we can, as larger as we can, and that still missing heritability would likely still be there, but it's not known at this point in time how much we can shrink that, for example, by increasing, by using whole genome sequencing data, for example. And I will touch on that in this on the final part of the lecture. So now I'm going to talk about some of the biases in heritability estimates. So I've decided to focus on 3 biases. One caused by shared environmental effects, another one caused by population stratification, and finally one bias which we'll I think contemplate in trying to solve a bit more extensively during the practical, and that bias is due to the distribution of math and linkage disequilibrium of SNPs used in the GRM. So starting with shared environmental effect, I just want to start by underlining that phenotypic resemblance between relatives is not all due to genetic factors, right? Genetic factors do play a role, but it's not just that. And so, in a way, remember this equation that I had, which I've shown multiple times in the previous parts, that the phenotypic correlation between relatives was linear function of the genetic relatedness, and we were sort of leveraging that to estimate heritability. And there is another part, there is a residual there. And that residual contains things that may not be genetic per se, but to how much the non-genetic factor vary with genetic relatedness is unknown. And if that residual part is correlated with the genetic effect, then that will create a bias. So there are different ways we can diagnose that bias. And one possible way is to look at estimates of heritability based on different combinations of the related relatives types we have in the sample. So we can, for example, run one analysis with MZ or monozygotic twins and DZ dizygotic twins, and just get an estimate of heritability. We can then shuffle and look at parent offspring and full SIBs, or avuncular and MZ twins, etc. And so with all these combinations, if we have no sort of violation, if we have no shared environmental effect, and I'm making another assumption, which I will talk about at the last part of this lecture, but in particular, if we have no non-additive effect, just to name that assumption, then we should have a consistent estimate regardless of how many combinations of relatives we use. And so if we detect that certain combinations create different estimates, that may be a way to diagnose the effect of some biases, I will say, in heritability estimates. So one possible solution is to model this shared common environment, and by modeling I mean making assumptions about it. And for example, one very common assumption is to assume that the degree of a shared environmental effect is similar between MZ twins and DZ twins. And if that's true, then we can have a consistent or unbiased estimator of heritability by looking at twice the correlation, difference in correlation between MZ twins and DZ twins. So this is one possible solution, which comes from making an assumption. And there is another solution, which is a bit more conservative, that says that, well, we can only restrict the analysis to people who are distantly related, because the likelihood of those people sharing a common environment is diminishingly small. So that could be a way. And so in the literature, people have proposed different threshold on the GRM to determine sets of unrelated individuals. The commonly used is 0.05, 5%, but you can be more stringent. Just if you want to really minimize that bias, you can take it to be 0.025. So another bias is created by population stratification. And so by population stratification, I mean the conjunction of two things. So on the one hand, we have genetic similarity between people who live in the same geographical area. And that can be explained by demographic history, the fact that genes travels with people, and people settling in certain parts of the world will set up their genetic variants there, and that will kind of create a more homogeneous genetic background for people living in certain geographical areas. And so, and on top of that, if you also have more similarity because of environment and the fact that people will tend to eat similar things, and that will show up, create a signal which is very similar to the heritability signal, because you have people with genetic similarity, increased genetic similarities, and as well increased phenotypic similarity. So if your sample is heterogeneous in terms of say geographical origin, it could be within continents, it could be across continents. And if that's the case, that will create an inflation in heritability, because it will say, oh, those people tend to be more genetically similar, and they also more phenotypically similar, and therefore the heritability is higher. So if those differences are for samples from, say, across continents, so what I call ancestry differences, although ancestry is we can have ancestry differences within a continent. But if we're talking about continental differences, where those differences are actually super large, the safest solution is to restrict your analysis to homogeneous samples in terms of ancestry. So for example, if you have samples from European ancestry and East Asian ancestry, you would like to analyze those samples separately and have an heritability estimate for those two samples, which can make sense in its own right, because as I said in part one, the heritability is not a universal constant. So there are potential true differences between heritability of those participants or individuals coming from different ancestries. And if there's the degree of stratification of sort of genetic difference between individuals in the sample because of ancestral background, if that degree is not that strong. So for example, what I'm illustrating here on the right-hand side of this slide is a principal component analysis based on the genotypes of populations in Europe. And so within continent, we can typically use those principal components as fixed effect in our model, just like those experimental variables I was talking about in the fixed effect in the part 3, we can use them in the model as a way to correct for those effects. So that's a very very powerful solution. So now I will turn to a third bias, which has been recognized, I think in 2013 in the paper by Speed and colleague published in the American Journal of Human Genetics. And in that paper, they were revisiting a number of assumptions underlying the genome model. And so, for example, there is the assumption that the SNP effects are independent, that they are normally distributed, that each of them contribute the same, and they played around with different scenarios for testing the robustness of the model. And by and large, the conclusion was that estimation of SNP-based heritability with GRML is pretty robust to a number of violations of its assumptions. But they also emphasized one particular violation that can create a large and substantially large bias, and that violation was essentially the fact that the distribution, the MAF and LD distribution of SNPs in the GRM may be very distinct from the one of causal variance. And when that happens, it creates biases that that can be significant. So in that paper, in Statistical 2012, they proposed a solution to that problem. And the solution was to actually model the SNP effects to create a dependency between the SNP effects directly with linkage disequilibrium. In that initial paper, it was mostly with linkage disequilibrium. It has been subsequently extended to create a dependency between SNP effects, in particular the variance of the UIs, so which I introduced in part 3, with a dependency between that variance and minor allele frequency. And so I'm just here listed a few more papers, in particular Singh et al. 2018, which looked at that using Bayesian models. And there are actually a number of method out there to essentially introduce those models and this dependency, between the variance of the SNP effects and LD and MAF attributes. But another method, which is a bit more agnostic, and this is the one I will talk about a bit more in the next slide, and the ones also we will be playing with during the practical. And this other method is more, I would say, it's non-parametric in a way. It acknowledges that there is a heterogeneity between causal variants, which are largely not observed, and SNPs used to infer heritability. And the way they decided to minimize that heterogeneity was by creating groups of SNPs that are more homogeneous with respect to certain MAF and LD properties. And so that has been introduced in Yang 2015 and refined and extended and compared with other method in Evans 2018. So just in brief, this sort of LD and MAF stratified method, or LDMS method, it goes like this. So the first step is to calculate the SNP attributes that we want to stratify the SNP on. So the first one is the MAF. So we calculate the MAF of the SNP, and we calculate another quantity, which is called the LD score. And that quantity will be instrumental in the lecture given by my colleague Hilary Funicane, who will talk about method for estimated heritability using GWAS summary statistics. And so this LD score is defined as, so it's a quantity for all the SNPs, and it's defined as the sum for all this, all pairwise correlation between that focal SNP and all the SNPs we have assayed, or even if we have in certain database. And so in practice, because of properties of the linkage disequilibrium, we actually don't calculate the pairwise correlation with all the SNPs on the genome. We actually restrict them to localized the vicinity around that SNP. So once we have those attributes, MAF and LD scores, then we create groups of SNPs that are homogeneous. For example, we can create 6 group of minor allele frequency from 1 to 5%, 5% to 10%, 10 to 20, etc. And then within each of those groups, we can then group together SNPs based on similarity on LD scores, and for example, using quartiles of the LD score distribution within those MAF groups. And so in this case, by intersecting those MAF and LD groups, we will have essentially 24 groups of SNPs, which are within groups are very more homogeneous in terms of LD and MAF than the set of all the set of SNPs as it was defined previously. And so then what we do with those groups is that for each of those groups, we can calculate a GRM. And essentially, we are turning our initial problem of looking at relatedness across the genome as one quantity. Now we have going to have 24 quantities reflecting relatedness based on SNPs having a certain property. And so then what we do is that we can actually extend the GRML model or the HE regression model. It works as well for both estimators. We can extend those approaches to fit jointly all those GRMs. So for HE regression, that will be turning to this sort of simple linear regression into a multiple linear regression, where we'll regress our cross product of standardized phenotype onto multiple values of relatedness. And for REML, that will be what I've shown here in this equation, is that we're extending this model where the variance covariance of the phenotype is in fact the sum across all those classes from 1 to k, and we have this k GRMs, and we get k variance components here to estimate. And so once we have all those estimates, we can have a genome-wide estimator by simply summing all those per class estimator of genetic variance. And what those studies have shown, the Yang et al. And Evans et al., is that this strategy produces an unbiased estimator of heritability if the grouping is refined enough, right? So there is a trade-off in terms of having too many classes, because too many classes reduces the biases. Because we're getting closer and closer, we have less and less heterogeneity, so it actually works for us. But what we lose is that by increasing the number of classes, we also increase the standard errors of our estimates. So there is a trade-off here in terms of precision and bias, which is a general issue in statistics. But this method overall works well without making again, any assumption whatsoever about the actual relationship, the functional relationship between SNPs and SNP effects and LD and MAF. So that's a nice property. Okay, so just to summarize this part, I've shown that a shared environmental effect in population stratification can bias heritability estimate, and the ways to fix that is to assess the genetic composition of the sample, for example, with genetic principal components, which is something you would have heard and you probably cover in the GWAS lecture about quality control. And so that's one thing important to do. And once we have those pieces, ideally recalculated in a more homogeneous sample, we can use them as fixed effect to correct for potential biases due to population stratification. And for the shared environmental effect, the most conservative solution is to only restrict the analysis to unrelated individuals. And so secondly, I've shown that there is another type of bias which comes from essentially a mismatch between the LD and MAF distribution of SNPs in the GRM as opposed to the causal variance. And there are different solutions, including modeling that relationship as has been shown in the literature, or by using Bayesian models, which actually select the variants that are most informative and thereby reduce the heterogeneity. And we can also use the LDMS strategy, which I expanded a bit more because we will be playing with that method in the practical. So I thank you very much for your attention. And I will expand a bit more in those practicals, and I hope to see you in the last part of this lecture. Thank you very much.